We wish to thank the Inter-American Tropical Tuna Commission for this invitation to participate in the Forum for South Pacific Swordfish. I am Jaime Alavarado Bremer, the head of the Molecular Ecology and Fisheries Genomics Lab, MEFGen, in the Department of Marine Biology at Texas A&M University at Galveston. This presentation will provide an overview of previous genetic work on Pacific swordfish population structure, and we will present the most recent advances on this topic using genomic data conducted by my doctoral student, Len Aguila. This research is a collaboration between TAMOG and the Pfleger Institute of Environmental Research, PEER, with the participation of Dr. Chugey Sepulveda and Scott Aalbers, and consists of the genomic characterization of swordfish tag electronically by PEER, which allows for the first time the direct comparison of satellite tracks and the genomic signal to delineate the potential stock boundaries of these highly migratory species. I will now let Len take the lead of this presentation.
Thank you for the introduction, Dr. Alavarado, and I would like to thank the IATTC for this wonderful opportunity to present our study. In this presentation, we will be looking at the prospect of a genomic approach in tandem with geospatial information in resolving the population structure of the Pacific swordfish. As an overview, we will be looking at the various Pacific swordfish stock hypotheses primarily based on fisheries data, followed by the different models of genetic population structure based upon past studies using a variety of markers and approaches. We will also briefly look at the genomic approach that we use in the lab, including the sampling requirements it entails, as well as some initial findings on our swordfish data. Over the last 40 years, numerous models of Pacific swordfish population structure based on fisheries data have been advanced. Some of these were summarized by Hinton and Alavarado Bremer in 2007 and vary in the number and location of subdivisions between putative stocks. To facilitate the visualization of different models, we can group them according to the number of stocks that subdivide the Pacific Ocean. Here we have two models of the two-stock hypothesis. The first model, which delineates the North Pacific stock from the South Pacific, was suggested by Nakano in 1998. Meanwhile, a second model, suggested by Ichinokawa and Brodak in 2008, assumes that this north and south delineation is right at the equator. Next, we have four models of the three-stock hypothesis. The first model by Sakagawa and Bell in 1980 subdivides the Pacific stock into North Pacific, Southwest Pacific, and East Central and South Pacific. The second model proposed by Barter and Cohn in 1988 delineates the Pacific stocks into Western and Central North Pacific, Southwest Pacific, and Eastern Pacific. The third model proposed by Nakano in 1998 divides the Pacific stocks into North Pacific, Southwest Pacific, and Southeast Pacific. And last but not the least, the fourth model suggested by Ichinokawa and Brodak in 2008 consists of a diagonal boundary between Western and Central North Pacific and Eastern Pacific stocks and a third South Pacific stock. This model is currently used by the ISC to define the management units for North Pacific swordfish stock assessment. Lastly, we have two models of the four-stock hypothesis suggested by Sosa Nishizaki and Shimizu in 1991. Both models present four stocks corresponding to Northwest Pacific, Southwest Pacific, Northeast Pacific, and Southeast Pacific, differing only in their respective stock boundaries. The second model was further supported by Hinton and DeRiso in 1998 and Hinton in 2003, as well as genetic studies by Reeb et al. In 2000 and Alavarado Bremer et al. In 2006. In addition to catch data, Fish population structure may be characterized by its gene pool using various data such as allozyme, mitochondrial, and nuclear DNA data. So far, there have been 9 published studies addressing the genetic population structure of the Pacific swordfish. 3 of these show genetic differentiation, and these are Grijalva-Chung's allozyme comparison of Hawaii and Mexico, the U-shaped isolation by distance corridor of Reeb and others with mitochondrial DNA, and the differentiation of the Eastern Pacific Ocean and the Southeastern Pacific from 3 Hawaiian samples using one locus, the LDHA. Recently, Liu and others published a comparison of samples collected throughout the Pacific Basin with 10 nuclear loci. Liu et al. Characterized swordfish samples throughout the Pacific Basin using high-resolution melting and found significant genetic differentiation among samples. The slide shows in bars the results of structure analyses where individuals were assigned to clusters, and in contours the inference by GeneLab using georeferenced individual genotypes. In both analyses, the nature of the geographic association of differentiation is complex and was summarized as differences between temperate and tropical regions. Such signal of genetic heterogeneity between tropical and temperate areas can be visualized in more detail in the structure results at k=4 clusters, with Northern Atlantic and Mediterranean samples included as outgroup clusters to better contrast the genetic signal of differentiation. In this figure, the blue and yellow signals represent the outgroups Northern Atlantic and Mediterranean clusters. On the other hand, the green signals depict the putative tropical cluster, while the pink signals correspond to putative antitropical cluster. The tropical and antitropical signals of differentiation were also contrasted using principal coordinate analyses to support structures cluster assignment. This study raises several questions, including their geographic boundaries, which demands for additional sampling, including early life stage samples. It also begs the question of whether these differences are not associated with sex bias dispersal of the swordfish. Although previous studies were able to find genetic differentiation and even discriminate these tropical and antitropical genetic signals with just 10 loci, this proved to be insufficient to resolve the population structure of swordfish in the whole Pacific Ocean basin. In addition to sampling coverage, the signal of differentiation should be tested with a large panel of nuclear markers for better resolution. This means a need to have a random representation of markers covering the entire genome, and this may be attainable by employing a reduced representation approach with the use of high-throughput next-generation sequencing. For my dissertation work, I am using dd-rad sequencing to characterize thousands of nuclear markers in swordfish to generate a random representation of genetic variations throughout the entire genome. This is in the form of single nucleotide polymorphisms or SNPs. However, this technology has the drawback of all other NGS approaches in that it requires very high-quality DNA samples. High-quality DNA samples are only collected from fresh samples. That's why I can't emphasize this enough. Tissue sampling is very crucial, and we need fresh samples. So what works? As I mentioned, we need fresh samples, and this must be obtained from fish that are still alive when they get on board. For us, we prefer fin clips over muscle tissues, and fin clips must be kept in ethanol and then refrigerated upon sampling and must be kept cold while in transport. As far as DNA extractions are concerned, these fresh samples provide us with excellent high molecular DNA that we need for massive parallel sequencing. Using this approach, our collaborators from Pierre have provided us with tissue samples from more than 100 swordfish that are tagged electronically off the California coast. And some of these swordfish have been characterized with dd-rad. With dd-rad, we were able to use about 48,000 SNPs across about 65,000 loci to do an initial cluster assignment of 115 swordfish. 5 of which are Northern Atlantic samples that serve as an outgroup, and the rest are samples from the Southern California coast and from north and south of Hawaii. The comparison of swordfish tagged from Southern California against the samples south of Hawaii that include juveniles provided by Robert Humphreys revealed that the Southern California samples are not homogeneous. To further understand the nature of the Southern California heterogeneous signal, we looked into the satellite tracking data provided by Pierre for those swordfish. This map summarizes the geolocations of tag deployment and reporting locations of 62 swordfish released from the Southern California bite. The availability of these geolocations presents a unique advantage towards the analysis of our genetic data. And accordingly, cluster assignment was rerun using the admixture software, accounting for this geographic information in our prior grouping. And the results are striking. When the location of final reporting for the satellite tracks is included, the geographic association with the genetic signal of differentiation of the samples from Southern California may be explained. These results underline the power of this approach of uniting electronic tagging with tissue sampling for genomic analysis supported by the California Sea Grant. Accordingly, the DD-RED approach that we are using holds a substantial promise to finally help solve, along with satellite tags and fisheries data, the swordfish population structure in the Pacific Basin beyond the EPO. Additional samples containing high molecular weight DNA are needed, and this would require the collaboration with scientists throughout the entire basin from many nations that we hope to get the privilege of having through this workshop. And finally, we would like to acknowledge the following people and institutions in helping us in the conduct of this study. Dr. Chugey Sepulveda and Scott Aalbers of the Pfleger Institute of Environmental Research, and Bob Humphreys of NOAA for providing us our much-needed samples for this project. The Pfleger Institute of Environmental Research, the California Sea Grant, the Fulbright Foreign Student Program, and the TAMAG Research and Graduate Studies Office for the research and student support, and the members of the Molecular Ecology and Fisheries Genetics Lab, Janelle and Joel, Erica, and Gio for logistical, computational, bioinformatic, and emotional support in between. Thank you for listening.